For my third lecture, I decided to do Chapter 4, Bacteria and Archaea. So let's get into it. So I decided to start off by talking about the structures of bacteria. The first structure I want to talk about is the pilus. This is required for conjugation, which is basically the reproduction of the cell. They use this to kind of latch on to other cells. They contain plasmid, which holds the DNA molecule of that particular cell. They have ribosomes. This is what translates the messenger RNA into the amino acid chains. Cytoplasm, this is where cell growth and replication occur. They also contain a cytoplasmic membrane. This helps protect the cell from any foreign objects, anything that would kind of damage a cell. They have a cell wall, and this helps maintain shape and structure so the cell doesn't kind of collapse on itself. A capsule, which covers the entire cell. This is sometimes like that slime coating. A nucleoid, which contains the genetic material of that particular cell, and flagellum, which provides motility. This is what helps the cell kind of swim around and move around. Next, we'll talk about the shapes of bacteria, and in the next couple of slides, we kind of get a little more into that, a little more in depth. But you have Bacillus, Coccus, and Spirillum. Bacillus is rod-shaped, Coccus is spherical shaped, kind of like circle, and Spirillum is a spiral, kind of like a squiggly line. So I provided some pictures on this slide so you can kind of get an idea. So you see that bacilli is little rods, kind of like, looks like tubes. Cocci is the circles, the spherical shape. And spirilla is the spiral shape, kind of like I said, a little squiggly line. Those ones are kind of easy to remember because it's kind of in the name. So we'll talk about bacillus or bacilli first. And as I said, these are your rod-shaped bacteria, kind of look like little cylinders. They are a gram-positive bacteria. They are aerobic, which means they do need oxygen. This type of bacteria contains endospores, and what these do are in unfavorable environments, they kind of help them survive, so it makes them more heat resistant or resistant to other destructive agents. I thought a little fun fact was that this type of bacteria, Bacillus cereus, it's a common soil bacterium that is found in raw milk. Next, we'll talk about the coccus or cocci. These are the sphere-shaped, which you think of circles. They're gram-positive or gram-negative, depending on the type. They're anaerobic, which means they do not require oxygen. They're kind of organized differently, and it determines the name. We'll go a little more in depth in that in just a second. But you have diplococci, streptococci, staphylococci. Streptococcus is an example of a coccus bacteria. This causes throat infections. This is the thing it seems all kids get when they start school back. Next we'll talk about spirillum or spirilla. These are spiral in shape, the little squiggly lines. They're gram-negative. These are microaerobic and aerobic, again needing the oxygen. They're the largest species of bacteria, and they're huge when you compare them to the size of other bacteria. H. pylori, this is a bacteria found in the stomach. It causes a lot of stomach issues, but this is a type of a spirilla bacteria. And these are those bacterial arrangements I was talking about earlier. So you have cocci, and as you can see, it's just one single cell. Diplo is where you have the two cells, they're kind of together. And then staph is a cluster of the cells, and it kind of looks like a cluster of grapes. And then you have strep, which is kind of stretched out in a long chain. I kind of think this looks like a caterpillar. That's kind of ways I remember. Next, we'll talk about gram-positive and gram-negative bacteria. So the gram-positive bacteria, they have a thick cell wall. They can cause your infections ranging from food poisoning all the way up to respiratory diseases. These are typically stronger than gram-negative cell walls because of the amount of peptidoglycan in the cell walls. Some examples of gram-positive bacteria would be staph, strep, and C. diff. I provided a little slide and it shows, you can see it, that they typically stain this purple color. And then you have gram-negative, and these are your bacteria that have a high resistance to antibiotics, which is really bad. They're a lot harder to kill out and get rid of. They do not contain the crystal violet stain, and they acquire genes from other bacteria that have become resistant. Their genes do mutate, and some examples of these are E. coli, salmonella, typhoid fever, the bad stuff. As you can see on the slide to the right, they kind of stain a reddish, pinkish color. Next, we'll go into sporulation and germination. So sporulation happens when a cell is introduced to an unfavorable environment for growth. So maybe it's too hot for that cell and it likes it cold. Well, when this happens, the internal and external of the parts of the cell signal the vegetative cell to cease growth. This kind of helps the cell survive in nutrient poor conditions. And when this cell enters a state that it can survive, this triggers the germination phase. This is when the cell kind of pops back to life and can reproduce and go on because it's now in a good nutrient environment where it can survive. Archaea and bacteria have well, they mostly have a lot of similarities, just a few differences. Bacteria was once actually defined as archaea, but they kind of ruled that out. So they are both prokaryote cells. Neither contain a nucleus or any membrane-bound organelles. They both have a cell wall and require their flagella to kind of move about. Um, the differences, archaea have no peptidoglycan in their cell wall, and they're more like eukarya cells, to where bacteria does have that peptidoglycan in their cell wall, but they're different than eukarya cells. And next we'll have the Burgess manuals. These are things that help aid in determining the type of bacteria. So the systematic bacteriology determines the identity of the organisms by emphasizing some of those characterizing aspects, to where the determinative bacteriology manual aids in the identification of bacteria, and they are divided into 35 groups.